Hello everyone, welcome to the new video tutorial of BioNTech. In the last session we have dealt about secretory tissue system. In this session we will be dealing with the shoot apex organization. If you haven't subscribed to our channel, subscribe to it and enable the bell icon to get daily notification. So without wasting time, let's get on with the video. Okay. So theories of shoot apex organization. So we have to learn how tissues are organized in shoot apex. To understand that, almost 6 concepts are there which were put forward by different scientists. So first 3 are very common, which we have learned in our lower classes. That is, the first one is apical cell theory, then histamine theory, and Tunica corpus theory, which is really important regarding the shoot apex organization, how tissues are organized in shoot apex. Then comes the mantle core concept, which is actually a modification of Tunica corpus theory. The name is different due to different scientists have put forward to it. And the 5th one is concept of French school, which is a really old one, and the Newman concept. So let's first focus on apical cell theory. The first one, that is the apical cell theory. Now this apical cell theory was brought by Nageli, which is a very common name. And then the important point that you have to focus here is that a single apical cell is a structural and functional unit which governs the growth of shoot apex. So that is what the theory says. So a single apical cell is there which governs the apical growth or which helps in the development of other further tissues after the apical growth. So that is the structural and functional unit of the shoot apex, or maybe root apex also the same theory comes. So similar there also. So a single apical cell is a structural and functional unit which governs the apical growth of shoot apex, and also helps in the formation of further tissues. Now the main important point, another point, is that it is found in cryptograms and not in sphenograms. So sphenograms, this is not found. This theory is applicable to cryptograms. Cryptograms are plants which reproduce by spores. So in pteridophytes and all this, the apical theory is what happens, what takes place in the shoot apex. So that's all about apical cell theory. Now let's focus on to the second one. Now the next one, that is histamine theory. According to this theory, there are 3 distinct meristematic zones in shoot apex. The first one is termed as dermatogen, the second one is peribulum, and the third one is pleurum. So these 3 distinct meristematic zones helps in the formation of further permanent tissues in the shoot apex of a plant. So what happens is that dermatogen is a meristematic zone which develops into the epidermis of a shoot. Then the peribulum is the meristematic zone, the next one, which grows and develops into cortex. The next one is pleurum, the central mass which grows and differentiates into steel. That is, the vascular tissues are formed from pleurum. So that's what this theory is saying about. There are mainly 3 distinct meristematic zones. That is the important point. And the names of the 3 distinct meristematic zones should be well understood. The first one is dermatogen, the second one is peribulum, and third one is pleurum, and what it forms, that also should be noted. That is epidermis, cortex, and steel. So that's all we have to focus on histamine theory. Now let's focus on the third one. Okay. Now the Tunica corpus theory. This is the most well accepted theory in the shoot apex organization. Although histamine theory is also a scientifically well accepted, there was one drawback of histamine theory. Then that is, in that theory there is mentioning of 3 distinct zones, that is dermatogen, peribulum, and pleurum. These 3 distinct meristematic zones are not clearly visible in gymnosperms and angiosperms, and that is why that theory was rejected. Now the Tunica corpus theory proposed by Schmidt. According to this, 2 zones are present, in which the first one is tunica, which forms the outermost zone, and it is formed by the anticlinal division. Anticlinal division means, suppose it's a cell and the next cell division is perpendicular to the first cell. So like this, a cell division occurs. Whereas in the case of periclinal division, the cell is like this, the next cell division is also parallel to this. So stacking of cell happens in periclinal division, whereas it does not happen in anticlinal division. So that's the difference. So in Tunica, the division is anticlinal, and the zone this outermost zone, which consists of 1 or more layers and is protective in function. The second zone is corpus, is a central zone which is protected by the tunica, and it is formed by a regular division, that is maybe periclinal or anticlinal. So it is not a regular division, it is an irregular division. So that is the 2 zones and what we have to focus on Tunica corpus theory. Now let's focus on the 4th one. Okay, now the 4th one is mantle core concept. It is actually relatable to the Tunica corpus theory, but the name is different due to a different scientist was proposed. So the scientist who performed is Profam and Chan, and it is relatable to Tunica corpus theory. And in this one, it's similar to the Tunica corpus, the name only is different. So the mantle is actually the tunica and the core is actually corpus. So it is the mantle forms the outermost zone and the core forms the central zone, just like tunica and corpus. So that's all we have to focus on mantle core concept. I'm just mentioning this theory because sometimes in the entrance and also this name might be asked from that portion, so you can relate it to this two-apex organization. So that is why I'm mentioning mantle core concept here. So now let's focus on the fifth one. Now the fourth one is concept reference school, which is actually an old concept. According to the three zones, just similar to the histamine theory. The naming also is in the French type. That is, the first one is anu initial, the second one is meristeme attente, and the third one is meristeme medullaire. So it's just like histamine theory, though the anu initial is the meaning is the peripheral active zone. So in the case of histamine theory, it was recalled as dermatogen. And the meristeme attente is next zone, which is inner to the peripheral active zone. In the histamine theory, it was recalled as peribellum, and the meristeme medullaire is central zone, which is also which is in the case of histamine theory, it was recalled as pleurome. So that is actually it recalls the histamine theory. So that's all we have to focus on concept of French school. Now let's focus on the final one, that is Newman concept. So finally, Newman concept, it is not actually a theory or something like that. He actually identified three types of initials in plants. The first one is monoplex, the second one is simplex, and third one is duplex. It is actually based on number of initials present. In monoplex type, there is actually one initial, so it is actually seen in vascular cryptograms and ferns. And in simplex type, maybe several groups of initials may be present, more than two types of initials may be present. It is actually seen in gymnosperms, whereas in the case of duplex type, it is actually seen in angiosperms, in which the three tiers of initials may be present, in which the outer two tiers may be tunica, and the central, then third tier is corpus. So like the tunica corpus organization is actually seen in angiosperm. So that's how he classified the three types of initials in plants. So that's all for today's video. If you have any doubt or confusions regarding this topic, please mail to bio@gmail.com. So wait till next video. Bye.